Thank you very much. Good morning. It's a pleasure to be here today, and we're looking forward to discussing this morning around the latest progress in drug development for Alzheimer's disease and dementias. To begin, I would like to hand over to our panel to introduce themselves, and we'll start from the end with Nicki, if that's okay.
Hello, I'm Nicki Johnson and I'm CEO of TRIMTECH Therapeutics, and TRIMTECH is focused on neurodegenerative diseases, developing small molecule degraders with CNS penetrance to target toxic aggregates. Good to see everyone.
Hello, I'm Kirsten Scott and I'm with AC Immune, which is a Swiss-based biotech. We have a number of assets in clinical development, including active immunotherapies, small molecules with intracellular targeting and PET tracers. And we have a number of partnered assets, but also wholly owned assets that are in development.
Good morning. I'm Gilles Beyen. I'm the CEO of Montis Bioscience. Montis is a company focused on targeting C1q, central node in neuroinflammation. So basically the topic here, what I will contribute is on neuroinflammation. Initially, we're targeting peripheral settings just because of the BBB, but working also to bring this important mechanism of action to the CNS.
So hello everybody, I'm Gabrielle Åhlberg Hillert. I'm the CMO at BioArctic. So my background is as a neurologist from the Karolinska, and there I have seen so many patients with these severe neurodegenerative diseases. So that's why I have a passion for developing new therapeutic alternatives. At BioArctic, we are actually the founders of lecanemab, Leqembi, now one of the first therapeutic alternatives for Alzheimer's disease, approved in over 50 countries. In our pipeline, we are focusing on toxic aggregates, monoclonal antibodies, but we do also have a proprietary BT brain transporter technology. And with that, we have a couple of partnerships with other companies such as Eisai, BMS, and Novartis. Thank you.
Good morning. I'm Alexander Gebauer. I'm the executive chairman and one of the co-founders of Galimedix. At Galimedix, we are working on small molecules that very well penetrate the blood-brain and the blood-retina barrier. Our target is amyloid beta, and we are modulating the aggregation process of misfolded amyloid beta species. We have several clinical assets now. We have a phase 2 program with eye drops running in geographic atrophy, and we just completed a phase 1 study with the oral formulation of the same drug, and we are planning to start an Alzheimer phase 2 study in probably a year from now.
Okay, so hello everyone. Thank you for being here. Thanks for the invitation to come. I'm Jenny Laird. I'm at Lilly, which is now 150 years old this year. So it's our big anniversary. But my role at Lilly is leading our neuroscience search and evaluation. So science technical side of business development, bringing things into the Lilly portfolio, all the way from very early stage academic collaborations out to large phase 3 acquisitions and partnering. So Lilly is focused on a range of neuroscience indications. Our kind of core interests are certainly in the topic of this panel, Alzheimer, dementia, and other protein misfolded disorders in neurodegenerative disease. But we also have very strong interest in a big portfolio in analgesia. And as we're kind of expanding our neuroscience footprint, we're also looking in very actively externally in psychiatry and other areas of neurology like seizure and movement disorders, etc. So looking forward to the panel.
And finally, my name's Emma Mead. I'm the Chief Scientific Officer at the Alzheimer's Research UK Oxford Drug Discovery Institute. We sit at the interface between academia and industry, and we take forward really promising and exciting new biology, translate that into drug discovery programs, and aim to partner those projects with industrial collaborators. And we also have a lot of spin-out activity as well. My background is in neurodegeneration and particularly cell biology. I've also worked at Lilly in the past, and I've been with the Oxford Drug Discovery Institute for about 8 years now. So I'll pass over to Jenny, who will kick off with the first questions.
Yep. So, the topic for our panel today is Alzheimer's disease and dementia. And so, actually in the panel earlier on this morning, mentioned that, the kind of second wave of innovation has already arrived. And Gabrielle also mentioned that the amyloid antibodies are now, launched. So they are a therapy on the marketplace. So one of the first topics we were going to think about was what this means from the perspective of co-pathologies. So, some things are now potentially standard of care in this field, which is exciting in itself for the first time, having something addressing the disease process. But how do we address this? Clearly, lots of people on the panel are working on targets beyond amyloid and tau. Some are working on amyloid and tau. So how are we going to handle the new era, new targets and co-pathologies? So I don't know if Nicki, you want to start from the...
So I think in terms of co-pathologies, the way I look at it is, yes, we have some efficacy with the amyloid antibodies, but that also reveals patients that don't necessarily respond. So we're working on tau, among other things. So the question for us is, what can we learn from both the responders but the non-responders to think, hopefully we'll get onto biomarkers. With the advent of biomarkers, does that really help us target down on those patient populations where combinations, or we can come in with our particular therapeutics? So I think we're going to learn a lot. We're also going to learn a lot from some of the partial responses that we're seeing with some of the other therapeutics as well, because that may be telling us something with anti-tau, for example. So for me, we're very early stage, so it's all about learning. It's all about really starting to understand what patients respond, what patients don't respond, and where we can really start to target those therapies down.
Keep going along the
line.
Yeah.
I guess I'd second those thoughts. And at AC Immune, we're also trying to develop diagnostics or PET tracers to facilitate the development of our assets so that we have a biomarker in order to select patient groups or to assess a treatment response. So we have a TDP-43 PET tracer, for example. Early preliminary results will be presented at ADPD in a few weeks, so I'm afraid I can't say anything. But we also have a TDP targeting therapies program, and I guess the idea is to try to get around the challenges of patient stratification by developing the diagnostic biomarkers or treatment response biomarkers alongside our therapeutic programs. Similarly, we have alpha-synuclein PET tracers. One of them works very well in MSA, and we're the second generation tracers under evaluation. I guess in the context of Alzheimer's disease, we know that alpha-synuclein co-pathology, TDP co-pathology is both important. And Gil is probably going to talk about neuroinflammation. But again, I guess it's another co-pathology that we think is important in neurodegenerative disease, not just Alzheimer's disease. And we have an NLRP3 inhibitor. I think it's earlier on in the field. It's a bit less clear, perhaps, what biomarkers and how to choose that patient population. And that's one of the challenges that we're looking at.
So what about neuroinflammation?
Yeah, so basically, and to your question on what next, before Montis, I'm now with Montis for 3 years. Before I was in oncology, and so I think a lot of parallels to be drawn from oncology, from indeed biomarkers and patient selection and combinations. Now, if you look at what's there today, and then indeed the link to neuroinflammation, for example, what we are focused on, obviously C1q, but C1q clearly. If you look at Alzheimer, if you look the fastest progressors in Alzheimer, and the dementia group in Amsterdam did a lot of work on this. Fastest progressors have very high C1q levels. Vascular dementia, very high C1q levels. Frontotemporal dementia, fastest progressors, very high C1q levels. So I think C1q on its own, as a biomarker, but also as a target, and what very uniquely, it's obviously there has been attempts targeting C1q as a complement inhibitor, but that's not enough. What Montis is doing is really targeting C1q as a complement inhibitor, but also as a microglia/macrophage reprogrammer with a dual mechanism. So I think there is clearly links to tau. There is links to NLRP3. So also early stage, as I said, we're sort of scratching the surface by starting in the peripheral settings, diseases like Guillain-Barré, CIDP. But basically, we have clear proof of concept. If we get the antibody into the brain, that targeting the dual mechanism in LPS models makes a big difference. But obviously, we need to get it there.
Yeah. So, Gabrielle, you still on amyloid or more new targets?
Well, I mean, I think amyloid is definitely at the center, but it, as a single pathology, it is rare. And that is the difficult thing for us. And we know that it's so much intermingled with all the other targets. At BioArctic, we are also working with alpha-synuclein antibodies, both naked antibodies and BT-associated. And we are now in early phase, or we are actually in the late phase 2A study, finishing in the study this year in Parkinson's disease and in MSA, multiple system atrophy, and then considering where to bring it from there. But of course, one could think, about Alzheimer's disease and alpha-syn pathology. They are also intermingled, as well as with tau, as well as with TDP-43. And I'm thinking there also about this today still neuropathology disease late, the limbic encephalopathy, that obviously I think, almost 50% of us will get it if we get over 80, 85. So there is something to think about more. So this is a very complex area, and today we only have one type of treatments, and that is for amyloid. We don't have any treatments for the other targets yet. No disease modifiers on the market yet, but a lot in development.
Yeah, so beyond antibodies, I'm guessing Alexander is going to...
Yeah. I think first I would like to jump back 20 or even more than 20 years. I think we are in a very, very exciting area in the field right now. 20 years ago, memantine was approved, and I had the pleasure of working at Merz at that time. And at that time, many clinical data were produced, and we learned a lot about the disease. And now, with the success of the antibodies, again, we have a huge wealth of data. I still think we need to better understand our patients, better stratify them, better understand who's progressing, why they are progressing, and what is happening beyond amyloid. But we should also look at the good data of the antibodies, but also at some limitations. I think amyloid has really been proven now as a relevant target in Alzheimer's disease. And we are working on it, as I said, in a different way. We do not have antibodies. We have small molecules that very well cross the blood-brain barrier. And we have seen effects beyond the classical anti-amyloid effect. We have seen effects on neuroinflammation. We have seen direct effects on morphology, and we have even in several preclinical models, I have to say, regain of function, sometimes even strikingly so. And so we're very excited to see how far we can translate that into human studies.
Okay, so I should ask the same question to Emma.
Yeah, there's so many exciting kind of sort of advances being made in the field at the moment, and I think that what we're seeing is a move toward looking a little bit more at earlier co-pathologies. So we see a lot of changes in neuroinflammation, but also early changes in vascular dementia or vascular changes in Alzheimer's disease. And I think that that's crucial to remember. We never really have a kind of pure amyloid patient population, as I think Gabrielle mentioned there. And I think it's important for us to consider how the disease progresses over time and think about how we can position our therapeutic strategies in the context of changes in things like the vasculature and the inflammatory processes that tend to precede a lot of the amyloid deposition. So for me, thinking about how these co-pathologies exist early in disease is very important when we consider targeting Alzheimer's as a continuum and a spectrum of pathologies.
And so can you kind of name any targets that your team are focused on specifically?
Yeah, we have a huge interest in particular in the TREM2 signalling cascade, as many researchers do, and we're watching with keen interest a lot of the TREM2 targeting antibodies and small molecule therapeutics, but also further downstream in that pathway, we have a particular focus on PLC gamma 2 as a differentiating strategy from TREM2, which is of real interest to us, and also other kind of inflammatory processes such as targets like galectin 3, which is clearly very important in mediating microglial activation. We're also particularly interested in considering how we can change brain blood flow, and that's very kind of a complex strategy where we're looking at sort of how we can impact the way that parasites are modulating brain blood flow in the context of their interactions with inflammatory microglia, for example, which we see changed early on in disease. And so that could be a very interesting source of new targets that we're quite heavily focused on investigating at the moment.
Yeah, okay. And I think just to kind of wrap up this topic, I'm wondering if any of the panel have any thoughts on how it changes clinical development if patients are already being treated with, or have been treated and perhaps stopped an existing therapy. So patients with amyloid cleared, for example, from their brain are perhaps different from, but so have you guys who are thinking about clinical trials been thinking how you handle that problem?
Well, I mean, I think at the moment it's a great opportunity because there's very unequal distribution of availability of therapies. So it offers the opportunity to stratify your trial by country because of the very different landscapes for treatment. It also makes things more complicated because you have to try and understand, do you give your treatment for co-pathology before, during, after? Where do you time it? Is it going to make it worse? Will it make it better? How do you get an adequate sample size in order to try and demonstrate that your co-pathology treatment is going to make a difference? So I think there's some challenges, but also I think opportunities for us as a field.
So I fully agree with you, Kirsten, but I think there is also another question to this, and that is really, if we consider neurodegenerative pathology, do we really think that we treat it and then we stop it? and that's a tricky one, isn't it?
Yeah, I would like to add on that. I agree with Gabrielle, and that might also open the opportunity for other approaches. And I think what would also be interesting to look at the combination of treatments. Now we have a new therapy that has been launched. We do not have data really now, and how could we, on how other drugs combined with the active treatment could. And we have some hints now. We do not have a full data set yet. That first, our drugs will not cause ARIA, but our drugs might have the potential of reducing ARIAs or the risk of developing ARIA with the antibodies. If that would fly, that could be a very, very interesting combination. You would have two drugs with similar, not identical, but having the same target, so related mode of actions, I would say, and the combination could 1 + 1 could make much more than 2. Could be interesting.
I think that's such an interesting point that both Alexandra and Gabrielle have made around different differentiating strategies, but also thinking about treating dementia on this continuum and considering this toolbox of therapies that we're going to need in the future and going forward is so vital. So yeah, really, really interesting comments there. Happy to move on and perhaps think about kind of how we treat patients. And as we've just discussed, we have now a range of potential therapies and different strategies on the horizon. And obviously the goal for all of us, I think, is to potentially try to intervene early and treat patients at the earliest time point and with a range of therapies across disease states. So really what we need to think about carefully is how we stratify our patients appropriately to make sure that we get clinical success with the therapies that are under development. And maybe I'd like to turn to our sort of more clinical colleagues first, so Kirsten and Gabrielle, and ask, what are the latest advances in things like biomarkers or also markers for sort of target engagement endpoints that might be important for us when we consider putting our therapies into humans for the first time?
So I mean, if I should think about something here, I'm of course thinking about the how to assess alpha-synuclein and the SAA assay, which is today, it is there, but it is sort of a yes or no answer. And of course, we would like to know much, much more about the quantitative and time-changing aspects there. And of course, we are waiting eagerly for the PET tracers for alpha-synuclein and for TDP-43. So, I mean, that's just one entry point to your question.
Yes, I think the seeding assay has been a really exciting development because it does allow us to identify people who are either positive or negative for alpha-synuclein quite early on. I think the hope with PET tracers, I guess, is also that might allow us to have a bit more quantification to allow us to measure the distribution and the spread of alpha-synuclein pathology. Because I guess the key question is, as we know from a number of these biomarkers, p-tau 217, seeding assay, or even things like GFAP in the context of Alzheimer's disease, that we can identify patients at risk many years before they're diagnosed. But the question we want to answer in terms of developing therapeutics is, when do we need to treat them? And ideally, we don't really want to treat people for 10 years potentially, but we want to have a clear idea of where the time to intervene is, where we get maximum response and we preserve the most function, but also we have drugs that are safe that can be used in those patient groups. And I guess the development of the fluid biomarkers, but also things like our PET tracers are helpful for that. I think in the context of alpha-synuclein, it will probably need to be a combination of things, because we might need different biomarkers to identify the pathology and to assess the treatment response. So I think we have enough data that potentially the seeding assay is not a good measure of treatment of response, but measures like having PET tracers over time will allow us to look a bit more at treatment response and try and understand whether impacting alpha-synuclein with treatments targeting alpha-synuclein actually have an impact in the brain, and whether that leads to clinical effects.
What will become a challenge in those things is also the safety, because the earlier we intervene, let's say for pre-symptomatic Alzheimer's, namely, the safer our drugs have to, because we would be exposing people for 5 years, 10 years, if one would intervene so early with the drug at a time when they have hardly any symptoms or no symptoms. So the drugs that we can use for those patients, they have to be very, very safe. That will also be one field that we have to look very closely.
Yeah, that's a really important point. And maybe if I could hand over to Gil and ask a question of that to sort of follow up. Obviously, inflammation is a very early hallmark of pathology in Alzheimer's disease and dementia. And I wonder if you have any thoughts on how we begin to find some of those very early biomarkers and measure the progression of those over the disease course.
Yeah, indeed, it's clear. As I mentioned, the link, the neuroinflammation as sort of the key indicator of the more. As far as I know, it's more the indicator of sort of the aggressive forms of Alzheimer or vascular. But basically, yes, you see already in the CSF C1q coming up early on. Now, C1q is complement and then linked to the safety. Do you want to already inhibit complement early on in the patient population? That's clearly a caveat and a question, basically. And so pushback we get from investors. Do you really want to target C1q early on? Basically, what our answer there is that we are, in fact, thanks to the dual mechanism of our antibody mainly focuses on microglia reprogramming first, because in fact, C1q has a complement function, but also has a very important complement-independent function, and that the complement-independent role, sort of microglia reprogramming, seems to happen at very low doses. The complement, much higher dose. So we're not there yet, but yes, it's we'll more work to be done there. Yeah.
Thank you. And maybe sort of shifting gears a little bit towards the tau biology, Nicki, you mentioned and you touched on in your sort of introduction there that, tau is also on a bit of a continuum, and I wonder how you've been approaching tau biomarkers or biomarkers of tauopathies in sort of across disease course.
So I think we're quite lucky with tau in that there are more biomarkers emerging. I don't think that's necessarily the case for some of the other targets that we're working on. So I think from our perspective, it's keeping an eye on the biomarker space as some of these therapeutics are starting to work their way through, both from a patient selection perspective at the beginning, but also if we start to see some efficacy with some of these therapeutics, what's happening to those biomarkers as well. So when you're early stage, you're in a quite privileged position in a way, in that you're able to learn from what's out there. But I do think tau is probably the most advanced when it comes to that. I guess a comment on sort of thinking about combination therapies again, and certainly as an early drug discoverer, I think what's quite interesting about this space is potentially how long people are going to be on these therapeutics as well. Obviously, we hope that people aren't going to be on them for too long. So measuring biomarkers and learning over time, very important, how the pathophysiology changes. Maybe if you intervene with one target, but you've still got other pathology going on. So I think it's not just necessarily about tau in my case, but actually what's happening to the other biomarkers as well. How can we make learnings? And I guess as I'm sitting listening to this as well, the other thing that I'm thinking is combinations. What can we learn from other therapeutic areas? Obviously, oncology, we talk a lot about inflammation. What do we learn from inflammatory disorders, and what does that mean for thinking about inflammatory approaches as well? So I know we started with tau, but I think there's so much to consider, particularly for early stage companies, because we're going to be watching others and taking learnings from that. And I guess one last point, as I think early, is yes, combination, but what do combinations with many, many modalities mean for patients, but also payers?
That's great, thank you. Jenny, maybe I'd just like to wrap up this question around biomarkers with you. Obviously, Lilly have had some huge successes in the field, and I think that is attributed to finding the right patient populations. So could you maybe comment a little bit about the strategies that have been used to identify and stratify patients for clinical trials?
Okay, so I guess we would probably attribute the successes of the amyloid antibodies to, finally, successes, because it took a long time, is to being able to segment patients with biomarkers. Firstly, to identify that they really do have the target of your therapy. So treating patients who actually have amyloid in their brain is definitely an advantage if you are aiming with an amyloid antibody. And that definitely, almost certainly wasn't true in some of the earlier therapy trials with earlier drugs. But also to being able to correlate the tau load, brain tau, to the sort of stage of progression, to be able to, so going from not just identifying people have the disease, but to stage of disease, and hopefully being able to predict at least a little bit speed of progression. And so, that's something that, not being an expert in oncology, but I do observe, is something that is now super standard in oncology. It's unthinkable of mixing stage 1 and stage 4 oncology patients in the same trial, whereas that is basically what we're still doing in many diseases in neurodegeneration, and hopefully that will change. And it's probably not just biomarkers that do it, it's also a sort of change in thinking. If you have got therapies, it certainly encourages clinical investigation and a lot more of the kind of natural history and observational studies to really identify what the course of a disease is and where you might be able to intervene. That's at least our thinking. But the big thing is moving from PET to blood-based biomarkers. That is going to change the world, hopefully, because PET is not a very accessible technology in a lot of the world. So what about you? We should ask you guys. From an academic perspective, How do you see developing a biomarker at the same time as a therapy? Is it even accessible?
It's something that we really strive to do at the Oxford Drug Discovery Institute. We recognise that we need to make sure that all of our targets are biomarker enabled, and this is a really important piece for us, piece of the puzzle really, when we think about partnering our programs. It's essential that we've got sort of projects that are end-to-end in terms of target validation all the way through to at least a strategy when we think about translating our research. And we use a lot of human iPSC technologies, specifically a lot of complex culture systems to try and balance and sort of partner those more target-driven biomarkers alongside the more kind of classical clinical biomarkers that we see. And that's our ambition. And it's a huge challenge and obviously expensive and time-consuming, but we really do hope to try and achieve that with all of our active programs at the moment.
So we're more than halfway through. I'm just wondering if there are any questions from the audience. I believe we have a roving microphone. So, but if folks would like to ask, do feel free. If not, we keep going. Ooh, you're not very lively this morning. I have to perk them all up. We have to ask you questions. That will get you going. Oh no, we do have one. There we go.
Thank you so much, Frauke Hein. I was just wondering, learning a lot about biomarker strategies and how you stratify patients. So I have not heard any word on machine learning AI or something like that. Complex clinical pathologies, tons of data, longitudinal studies. So for me, it would be great to hear if there's anything what you guys are considering to include that into your decision making.
Well, maybe I can start. I mean, I'm not at all an expert in this field, but evidently we have to follow it so closely. At BioArctic, we are not driving this field on our own in any way, but we are collaborating a lot. And one area I will just mention that is really, really dear to my heart is how we can create synthetic placebo arms so that when we enter patients into our trials, they, as few as possible, will need to be exposed to placebo. They can actually get the active treatment because we are able to create their digital twins when entering the trial. So that's something we are looking very much into, and we are also driving a TDP-43 ALS project. And of course, that's an area where I would like in the future to employ this. This was just my entry point to the topic.
Yeah, we are much earlier, but we are right now, we are designing a phase 2 program for Alzheimer's disease. And for the exactly same reason that Gabrielle mentioned, we are also thinking about applying AI for the digital twin concept that will reduce the number of placebo patients, hopefully, but also for patient selection. And defining responders, non-responders, trying to better understand the population, and then in later stage trials have larger effects because we have a more focused patient population. I think there are lots of things that need to be done and will be done. It will be possible in the next 2-3 years. I think it's a very exciting field. We are using heavily AI in our ophthalmology program. The ophtha guys seem to be a little bit more advanced than our field is.
And I think the challenge for small biotech companies is, I was at AstraZeneca before, where we had an AI department that was bigger than AC Immune entire staff. And so for biotech companies, trying to implement AI-driven strategies is hard. And I think what we're trying to do is build up collaborations with academics and other companies where we can leverage learning from AI, but also from patient cohorts. And my research background before I moved into industry was mainly in working with patient cohorts and how we can use that understanding of the natural history of disease. And now that we have much bigger data sets and the ability to look at them in depth with AI, I think we will get rich learning from that. So I think it's a great thing for all of us that will help running trials in the future.
We have 2 more questions.
Yeah.
Quick question. What is the latest data on the prevalence of AD, and does this data include India?
Good question, to which I do not know the answer.
I don't know the number, but I think there's some recent papers in the Lancet Neurology that have looked at global burden of disease. And there is indeed a huge burden of disease, not just in India, but also in sub-Saharan Africa. that I guess, those patients tend not to be included in our trials. And also they are going to be very last to get access to a lot of the therapies that we're working on. So I guess I'm not sure we have an answer to that, but I think it is an important massive population that remains it's a big unmet need in an area where there's a lot of unmet need.
I'd say we have to caution the difference between Alzheimer's disease and dementia, because measuring dementia is a clinical diagnosis, which, but dementia can have multiple causes. Defining Alzheimer's disease requires either access to one of the tests, preferably PET ligands, or post-mortem pathology. I'm not sure that, I honestly have no idea how prevalent those kind of technology or how available those technologies are in India, and fairly sure they're not very available in sub-Saharan Africa. So we may not know for a while until things change. Maybe we will find out, like we have with some other diseases, that you find out the prevalence in these lower economic or lower, less well-resourced healthcare systems when a cheap treatment becomes available and you see who is cured. Then you find out how many people actually had Alzheimer in those conditions.
And it's not only a question of technology access and diagnosis, it's also a cultural phenomenon. There are some cultures where the elderly are treated different than in the West, and where dementia is not even diagnosed, although people realize that something with grandfather or grandmother is not like it used to be. But culturally, one does not want to name this a disease. So there's probably a gigantic underdiagnosed field, even in this demographically young populations. Probably there are many, many patients that have dementia and Alzheimer's that even go unrecognized, I would say.
One more.
I was wondering how the companies on the panel here are assessing the changes at the FDA with regard to clinical trial design. The example of Unicure last week, where they were told, and I think this brings up also what Ms. Åhlberg has mentioned, the need for placebo trials too with severe disease like Alzheimer's. How have you seen the changes? Do you see it positive? Do you see it negative? Do you see it just complicating everything? What's your thoughts that you would want to share here with the audience?
Anyone want to take this?
I can have a go. I think my observations as a clinician is that the placebo effect is a big thing. So I think I understand the FDA's rationale for arguing that we still need the placebo effect. And there's plenty of examples of therapeutics where, many small phase 2 or open label studies showed a good effect. And then with a well-powered, well-designed phase 3 with a placebo group, it's confirmed that the drug has no effect. And I guess for that reason, I think until we have robust systems for digital twins or for looking at patient deviation from predicted trajectory, until we have validated replacements for a placebo group, it's very difficult to argue for not having a placebo, just because there's so many things that can be driven by that placebo response. In ways that we don't often capture. There's sometimes inadvertent unblinding, that both clinicians and patients want drugs to work, that, you look at areas like pain where you have a huge placebo response within the first few weeks of treatments with any therapies, but it also applies to neurodegeneration, where we've seen trials with ALS, with MSA, where there's early very positive results, and then subsequently in larger studies, they disappear. So I think until we have a better solution, we do need the placebo group to be really sure that we have a robust effect.
Okay, more questions? No? We will carry on with our questions then. Okay, so thinking about how do we convince people? I mean, the FDA being one of them, but it's not just evidence to convince the FDA when you finally get to a registration. Also, the sort of evidence that you might need to convince investors or, and as you go through the process of development, convincing patients that it's actually worth signing up for your particular therapy. So I wondered, again, we can go along the line, but equally we can have people jump in. What are the kind of things you think about in terms of presenting evidence for an investor to convince them that?
So I guess I can comment on this actually. I think with TRIMTECH, it was an interesting phenomenon compared to maybe other biotechs I've been in before. And that's because our focus is on targets that you can call hot or you can call people are convinced in, however you want to describe it. So tau, alpha-synuclein, Huntington, everyone immediately recognizes them. So either there's a genetic linkage or people believe already and there's a clear unmet need. So that actually, for a biotech, when you're looking for investors, is the most powerful argument that you can have. As you start to think about maybe adjacent pathophysiologies or mechanisms, then I think, it's more difficult because people need convincing. And then you really have to start to think about what are the potential in vitro and in vivo models where you can show efficacy that potentially investors or pharma believe in. So understanding at least what models people believe in and how you draw the mechanistic link to your disease is very, very important as well. So I guess I'm saying it really helps if people already believe, quite frankly, and that really is a thing. But other than that, you've really got to work hard on building the case for the linkage to the disease and take people on the journey through mechanism, through phenotype, through genetics, if you have it, through describing the disease. Biomarkers as well come into that. So a bit of a mixed bag, but it is harder if you're trying to pull in a new mechanism.
Maybe I can I fully agree with that. Working on a interesting target, but nobody really knowing C1q. With the last 2 years, we've been really doing heavy lifting in vitro, 6 or 7 in vitro studies, and it's only as we started to, and luckily we're a spin-off of the VIB, so we're seen as high science, so very solid science, but it has really taken to put different pieces of the puzzle together in 3 different models, model of Gebauer, model of MS, that people now start to see, yes, this indeed, this dual, first of all, C1q as a target increasingly is known, but then also that this dual mechanism can work, and basically that's where we think we can get the attention now from investors and pharma.
We try to not only build on the solid science. I think solid science, that's the basis. If you have weak data, then nobody will invest. But we also try to look at what do the patients need and what in the end will the payers expect. And hopefully that's going to convince the investors that we have the solid science, that the target product profile that we have defined is achievable, and the target product profile to be meaningful to the patients and ultimately to the payers, because otherwise you might have good science, but you might not have a commercially valid drug. So far, we have been quite successful in fundraising. That was driven by the OFTA part of our company. Now let's see whether we can convince investors into the Alzheimer's story as well.
So I'll take an example from reality, and didn't say that this was not reality. That was not what I meant. and ideally, of course, we should have the target genetically linked, and that is the case for alpha-synuclein to the alpha-synuclein disorder. So we know we are now with BioArctic, we are now in phase 2A in Parkinson and in MSA. Of course, this will be a study showing safety and tolerability. Is that enough? We know that next step, phase 2B, we could go into much broader, of course, motor Parkinson could be one step. Another could be multiple system atrophy, MSA. Another could be cognition in DLB or Parkinson dementia. All of these requiring quite large efforts, quite large undertakings for a company such as BioArctic. And ideally there, we should have the biomarkers super linked to the target and the indication. And we are not really there yet. So this is a very difficult and very realistic question you're posing to me, and I don't have the answer.
So we have 4 minutes left, and we'd just like to wrap up by asking our panel what the most exciting trial or event in the drug development landscape will be over the next year. So starting with Nicki, what are you most looking forward to seeing in the next 12 months?
I think, for us, with a focus on tau, again, I think it's the Biogen data will be really, really exciting to see that, particularly as that is an approach that when some of the antibody data has come out and potentially been quite disappointing in some cases, I am very focused on removing both intracellular and extracellular tau, and that has been the argument put forth, for example, by Biogen. And I'm sure there's many other things that can interfere with that clean result along the way, but I'll be very interested from an efficacy perspective to see that data coming out. And if I can have two shots on goal, because I saw Rich from Unicure actually a minute ago, I'd really be interested to see the Unicure journey actually, because although I think it was overstated to some degree in terms of excitement, that will be so meaningful for the whole field going forward.
I agree about intracellular targeting and tau. I guess I'm also interested in seeing the Lundbeck results from their MSA study with their alpha-synuclein targeting antibody, which I guess won't be this year, but I think that is will be an important result for the field, whether it's negative or positive. I think will be really important for those of us working on alpha-synuclein to learn from. And also looking at the NLRP3 inhibitors as they go through development. There's some results coming out from the Roche NLRP3 inhibitor in PD, which we're watching with interest, and Nodthera, who also have NLRP3 inhibitors in development. And we have our own in healthy volunteers this year. So we're hoping there'll be more development in that field as well.
I also was thinking of NLRP3, sort of neuroinflammation again, the Ventyx. I don't know when the data will come, but the next data that looked interesting, more in the C1q space. There is nothing really in CNS ongoing, but for example, the work that Argenix is doing in CIDP, C2 inhibitor, all of that I think we will learn, and for more for CNS. These are data, I think, also end of this year, maybe early next.
So I'll allow us to look a little more forward than just the next year, and that will, of course, be the pre-symptomatic Alzheimer's trials, the donanemab Trailblazer and the lecanemab AHEAD trial, and to see how they pan out. That's going to be so important and interesting, and see how that's going to be implemented in clinical practice. That is a difficult question.
Yeah, I'm interested to see, and this hopefully will happen over the next 12 months or so, whether tau, besides being a biomarker, is an actual drug target and a valid drug target. I'm not so fully convinced up now. Hopefully I'm wrong. And then we have another thing. Hopefully within a year, we should have the first hints of data from our running phase 2 study in ophthalmology. And if that is positive, at least we have shown that the whole principle would work. And that could be very, very important for us.
Both of mine got stolen, not stolen, but no, prevention. That is going to be so interesting. But I am also really interested in the tau. Of course, on the ASO siRNA possibilities in neurodegeneration. I think we should start to see some of that data this year.
And finally, I'm going to say that whilst disease-modifying therapies are incredibly important, I think we have to remember that there are patients who are living with debilitating symptoms that we need to target at the moment. And I'm very intrigued and excited about seeing how the clinical trials for some of the antipsychosis therapies, such as the BMS trial, will be received and read out in the next year. I think that'll be very important for patients who are in need now. So we are at the end of our panel. I'd like to just thank our panel members so much for their participation and their insights. It's been a really interesting discussion, so thank you, and thank you all for your attention. And I'm sure everyone will be around during the break for more discussion. Thank you.